Welcome. Today we are discussing enzymes, definition and classification. Definition and classification of enzyme can be asked as a short question or it can be a part of a long question on enzymes. तो enzymes को हम define कैसे करेंगे? Enzymes are biological catalysts or biocatalysts which are synthesized by living cells. They are mostly protein in nature with the example of RNA, which acts as ribosome. Enzymes are also colloidal, that is, they are water soluble. Enzymes are thermolabile, that is, if you heat or increase the temperature around 90-100 degrees, most of the enzymes are denatured. Most of the enzymes are active around 40 degrees Celsius, 35 to 40 degrees Celsius. Thermolabile with the example of Taq polymerase, which is used in polymerase chain reaction. It's an enzyme which is obtained from the bacterium Thermus aquaticus, which is found in hot springs, and that Taq polymerase is known to be stable or active even at temperatures around 90-95 degrees Celsius. And enzymes are highly specific in their reaction, in the sense that there are different reactions have specific enzymes which will catalyze that reaction. So how do you define enzymes? Enzymes are biological catalysts produced by living cells, mostly protein in nature with the exception of RNA, which acts as ribosome. They are colloidal, thermolabile and specific. So just to give a couple of minutes to understand the concept of enzyme. Enzymes, what is biocatalyst? That is, they increase the rate of the reaction. They increase the rate of the reaction thousands of times. Sometimes the reaction is possible on paper, but it may not be practically possible. Enzymes make it practically possible. There is an example given that a man had 17 horses and he wanted to distribute his 17 horses between his three sons such that the first son gets half the number of horses, second son gets one third the number of horses, and third son gets 1/9 the number of horses. If you divide the number 17 by 2, you get 8.5. By 3, you get 6. By 3, you get 5.66, 5.44 something. If you divide by 9, you get 1 point something. So if you divide it by 2, 3 or 9, you are not getting a whole number. And that is why the three sons couldn't divide the 17 horses according to this formula that the man wished. And they started fighting with each other. And then that is when one of the neighbor had a black horse. He brought the black horse into the fold. He said, you consider this black horse, and now the total number of horses becomes 18. And then now you divide the horses based on the formula. So the first son got half of 18, which is 9 horses. Second son got one third of 18, which is 6 horses. Third son got 1/9 of 18, which is 2 horses. So 9 plus 6 plus 2 is still 17. So the 17 horses of the person got divided between his two sons, and the one black horse went back to the neighbor. Enzymes are similar. Enzymes increase the rate of reaction several thousand folds, or you can say that if the molecules are left by themselves, the reaction may not happen or it may take a lot of time to happen, but enzymes make it happen in a few seconds. Reaction which is theoretically possible, enzymes make it practically possible. Like that black horse made division of 18 into half, one third and 1/9 practically possible, which was not possible with 17. But that black horse did not get divided to any of the three sons. He went back to the neighbor. Similarly, enzymes catalyze or increase the rate of the reaction, but they do not take part in the reaction. They do not get altered during the reaction. You can also call teachers as enzymes because a good teacher can catalyze the process of studies, and what you can understand by 1 hour of reading by yourself, maybe a teacher can make you understand in much lesser time. So that was about the definition of enzymes. Another thing is hydrolysis of proteins. Uh, all chemical reactions in the body are carried out by enzymes. And say hydrolysis of proteins, if a strong acid, if you boil it at 100 degrees, it would take several hours. But the proteolytic enzymes in our digestive tract, they digest the protein in few minutes. That is how powerful the enzymes are. And the study of enzymes is important because enzymes these days are a marker of function of of every organ, and they are also markers of several diseases. So study of enzymes also have therapeutic potential. Enzymes also have diagnostic potential. There is a lot of importance. That's why study of enzymes is important. This was about the concept of enzymes, and then we will go to classification of enzymes. So coming to the nomenclature and classification of enzymes, enzyme names usually end with the suffix ase, like urease, elastase, lipase, and that generally also tells you the reaction that it catalyzes or the molecule on which it acts, like urease acts on urea, lipase acts on lipids. But there are some exceptions, like there is trypsin, chymotrypsin, etc., that don't end with this suffix ase. In fact, if you see these proteolytic enzymes like trypsin, chymotrypsin, and some other enzymes in the body, they are secreted as inactive precursors, which are called as zymogens. Now, why are they secreted as inactive precursors? That is, if you take the proteolytic enzymes, trypsin and chymotrypsin, for example, if they are released in the exocrine pancreas where they are secreted, if they are activated at that site, that will cause autodigestion of the pancreas. And that is what happens in acute pancreatitis, that the enzymes get activated within the pancreas itself, causing autodigestion. And we don't want the enzymes to be active in the pancreas. We want the enzymes to be active in the small intestinal lumen, in the duodenum, where they will cause breakdown of proteins into amino acids as a part of digestion. That is why they are secreted as precursors. These zymogens usually have the prefix pro, like prothrombin, proelastase, not always, but sometimes. And they usually have suffix ogen, like chymotrypsinogen, trypsinogen, pepsinogen. Active forms will be called as chymotrypsin, trypsin, pepsin. So that is about the general casual nomenclature of the enzymes. So as you may have noticed that in the general nomenclature of enzyme, some enzymes end with ase, some don't end with ase, and the name of the enzyme does not always tell us a clear idea about the reaction catalyzed by the enzyme or the substrate on which it acts. There is no uniformity. There was no uniformity in the nomenclature of enzymes, and it was very haphazard. So, to make the nomenclature uniform and more organized, the International Union of Biochemistry, IUB, or you can also call it as the International Union of Biochemistry and Molecular Biology, IUBMB, set up an enzyme commission, which in 1961 came up with 6 classes of enzymes. In 2018, that enzyme commission added a seventh class, translocase. So currently we have 7 classes of enzymes. Rather, The reactions catalyzed by enzymes can be divided broadly into 7 categories, and the enzymes hence can be broadly put into 7 classes called as oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, and translocases. So you can remember this by the acronym ORTHLILT, right? Translocase was added in 2018. And as far as how to remember if lyase comes first or ligase comes first, if some of you are spiritual, uh, we can remember it this way: before we can connect to God, we have to break our attachment from all the worldly things, and then we can connect to God. Even God, when He wants to come close to us, most of the times He breaks our heart. So lysis happens first. पहले दिल टूटता है और फिर दिल भगवान से जुड़ता है. And so Othlilt, the first L is lyase, second L is ligase. First is lysis, second is ligase. And so this enzyme commission came up with something in chemistry we have heard of IUPAC nomenclature. Similarly, enzymes have a common name, like we have nicknames and we have official name. Enzymes have a common name, and enzymes have a IUBMB name. So the enzyme का name is written. It has 4 numbers. The first number uh, tells us the class of the enzyme, whether it is oxidoreductase, transferase, lyase, isomerase. So oxidoreductase would be 1, transferase would be 2, hydrolase would be 3, lyase would be 4, isomerase would be 5, ligase would be 6, and translocase would be 7. Second is the subclass, third is the sub-subclass, and fourth is the enzyme within that subclass, sub-subclass. To give an example, alcohol dehydrogenase is the name of the enzyme. The name suggests that it causes dehydrogenation or oxidation of alcohol into aldehyde, but its official name is alcohol NAD oxidoreductase because it belongs to the first class oxidoreductase. NAD acts as the coenzyme, and NAD also acts as the acceptor of the hydrogen. And the enzyme commission number of alcohol dehydrogenase is EC1111, where the first one represents oxidoreductase, second is the subclass, third is the sub-subclass, and alcohol dehydrogenase is the first enzyme of that sub-subclass. So this is how the enzymes are given the official name, enzyme commission name, and the enzyme commission number, which has 4 digits. Now we'll go to understanding the reactions catalyzed by enzymes of each of the 7 classes with examples, which you will also have to write as an answer in your exam. So coming to the understanding of each of the 7 classes of the enzymes, I will not go much into details. I will just stick to what you can write as a part of your answer in the exam. The first class is oxidoreductase. Oxidoreductase basically it oxidizes the substrate, and that is it catalyzes the oxidation reduction type of reaction. Oxidation is loss of electrons. Oxidation we'll also learn further in biological oxidation. Oxidation is loss of electrons or loss of protons or hydrogen atom, or Oxidation is uh, oxidoreductase is catalyze oxidation reduction type of reactions. Oxidoreductase catalyze oxidation reduction type of reactions. General reaction can be written as AH2 plus B, hydrogen goes to B, BH2, and A gets oxidized, AH2 gets oxidized to A. So example can be say glyceraldehyde 3-phosphate dehydrogenase of glycolysis. So you can write glyceraldehyde 3-phosphate, glyceraldehyde, glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate dehydrogenase converted to 1,3 bisphosphoglycinate, bisphosphoglycerate, which is also called as 1,3-BPG. And NAD acts as the coenzyme for this reaction. It also gets phosphorylated. Inorganic phosphorus comes from the matrix and it becomes 1,3-biphosphoglycerate, bisphosphoglycerate, and NAD gets reduced to NADH, and one proton is reduced into the is released into the matrix. Second example that you can give, is succinate dehydrogenase, conversion of succinate to fumarate as a part of TCA cycle. There are n number of examples, but you just have to write one or two examples as a part of your answer. So succinate dehydrogenase, as you all know, it's not just a single enzyme, it's a complex, succinate dehydrogenase complex. Succinate to fumarate, and FAD gets converted to FADH2. Succinate dehydrogenase complex is actually complex 2 of electron transport chain. Other examples that you can give, so when you write answer in the exam, you write oxidoreductases. It catalyzes oxidation reduction type of reaction. General reaction AH2 plus B is BH2 plus A. Then couple of examples with the substrate enzyme, the write a couple of reactions, and then you can write a couple of more enzyme names just as example. So you can write lactate dehydrogenase, alcohol dehydrogenase, which we took as an example in the previous slide, and say acyl CoA dehydrogenase. Second class of enzymes is transferases. It is very easy actually to remember the flow. There is a flow to this classification. Oxidoreductases, they catalyze oxidation reduction type of reaction. Basically, they catalyze transfer of protons or electrons and transfer of any other group other than hydrogen. Transfer of hydrogen. So oxidoreductase is transferring hydrogen from one compound to another. Transfer of any other group other than hydrogen from one substrate to another is done by transferase. So general reaction would be AX plus B, that X gets transferred to B, B becomes BX, AX becomes A. So example of this can be glucokinase, right? Glucokinase or hexokinase. So glucose to glucose 6-phosphate. I'm drawing these reactions purposely because when you study from the book, you should not just read the reactions. You should practice writing the reactions or writing the flow chart or the cycle with the substrate, the enzyme, the coenzyme, product, whatever is happening in the reaction. If you practice drawing those flow charts and cycles and pathways, then that will become muscle memory. You will remember it better rather than just reading. And that's why I'm taking I'm doing this thing where I'm writing as I explain. So glucose 6-phosphate, hexokinase or glucokinase. Glucokinase is in the liver, hexokinase is in all tissues, and ATP acts as a phosphate donor, and ATP becomes ADP. Magnesium acts as a cofactor. We'll learn very soon what are cofactors. And second example can be, say, norepinephrine to epinephrine. If you are new to biochemistry, obviously many of these enzymes you wouldn't know, many of these reactions you wouldn't know. But if you are studying it now as a preparation for your prelim exams or your university exams, you have covered almost entire syllabus. Most of the reactions you will have studied somewhere in some or the other chapter. So studying enzymes will become easier now than when you are studying enzyme as a newcomer in the first year MBBS, when you have to mug up most of these reactions and enzymes. But that's okay. So norepinephrine to epinephrine, methyl group is getting transferred here. So the enzyme name is methyl transferase. And S-adenosyl methionine or SAM is the most common methyl donor, or you can say almost the universal methyl donor in biology, favorite methyl donor. S-adenosine methionine gets converted to S-adenosine homocysteine by giving methyl group to norepinephrine, which becomes epinephrine. Other examples of transferase are phosphofructokinase. Which reaction does it catalyze? You have to read about it. Transaminase, phosphofructokinase also catalyzes a phosphorylation reaction, you know, from glycolysis. Transaminase, transaminase catalyze the transfer of amino group, amino group from an amino acid to keto acid, and the amino acid becomes keto acid becomes amino acid. So this was about transferase. The third class of enzymes is hydrolases, which catalyzes splitting of molecules by using water. Splitting or breakdown of molecules by using water is also called as hydrolysis. General reaction can be written as, if there is this molecule AB and you split it into two using water, so water will become H and OH. OH will go to one of them, H will go to one of them, so A can become AH, B can become BOH, or B can become BH, A can become AOH, depending on whatever the reaction is. So this could be written as the general reaction for hydrolases. AB plus H2O gives AH plus BOH. Example are lactase, urease, they are all hydrolases. So lactose being broken down to glucose and galactose is hydrolase enzyme lactase. Urease, so there is water, right? Lactase plus water is glucose galactose, by lactase. Urease, sorry, very sorry. Urea on hydrolysis with urease yields two molecules of ammonia. and carbon dioxide. As you know, formula for urea is CO NH2CO NH2, right? So on addition of water, it splits into two molecules of ammonia and carbon dioxide. There are other examples of hydrolysis like sucrase, maltase, etc., etc. Right? Fourth class of enzymes. Fourth class of enzymes is lyases, which breaks down molecules by mechanism other than hydrolysis, that is, without the use of water. So, if you want to remember OTH, OTH, oxidoreductase, transferase, hydrolase. So, what is the first L after hydrolase? Hydrolase is breakdown using water, breakdown without water, so it is lyase. First L is lyase. So lyases catalyze splitting of molecules by mechanism other than hydrolysis. Reaction can be written as AB plus XY. This becomes AX plus BY or AY plus BX, whatever you want to write it as. This can be the general reaction. Examples are fructose 1,6 biphosphate, either fructose 1,6 bisphosphate. What's the difference between biphosphate and bisphosphate? Something that you can read about and comment. Fructose 1,6 bisphosphate by aldolase. Aldolase converts into glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. Enzyme is aldolase. Pyruvate decarboxylase. So pyruvate it will cause acetaldehyde, breakdown of pyruvate into acetaldehyde plus CO2. So other examples of lyases can be aldolase B. Now this one, fructose 1,6 bisphosphate, this is aldolase A. Aldolase B is also a lyase. What reaction it catalyzes, that is your homework. And fumarase, that also is a TCA cycle enzyme. So fumarase, that also is an example of lyase.